This is Tarun, roll number 25, MSBT54, presenting on the topic Principle and Applications of Affinity Chromatography. Affinity chromatography is a powerful purification technique that separates biomolecule based on the interaction of target biomolecules with that of ligands. Such purification techniques are usually used to purify substances like enzymes, antibodies, and recombinant proteins. This technique was discovered in year 1968 by two scientists called Pedro and Mayer. The principle on which the affinity chromatography works is the ligands are immobilized on a solid matrix such as agarose or cellulose beads, to which the mixture or the mobile phase that contains the sample is being poured. The In the mobile phase, the target molecules have specific affinity towards these ligands and get bound. The rest unwanted molecules have been washed away, and later on, using a special elution buffer, the target will get separated, or by altering the environmental conditions such as pH, concentration of salt, or by introducing a competing ligand, we can separate the target molecule. The key steps involved in affinity chromatography are preparation of stationary phase, where we attach the ligand on a solid matrix. The ligand can be an antibody, a substrate, or a metal chelate. The second step would be the preparation of sample mixture, where we choose a target molecule that has a specific affinity towards the ligand. The third step, we separate the unwanted molecules by washing them to the column. The fourth step, which is important, where we use a special elution buffer to separate the target molecule. The advantages of using affinity chromatography is it is high. It has high specificity due to unique biological interactions such as enzyme and substrate activity and antigen antibody activity. It has the ability to obtain highly purified products in single step. Here we have an image explaining the working of affinity chromatography. This beaker contains the mobile phase which has a sample mixture which is poured to this column that has the agarose base, which is a stationary phase containing the ligands. The target molecules get bind to the ligands, whereas the unwanted molecules are washed away. Later on, using a special elution buffer, the target molecules are also separated and collected separately. Applications. Common applications are protein purification, enzyme isolation, where we do the immobilization of enzymes, and for separating clotting factors in blood. It is also used in detecting biomarkers during disease diagnosis and is also used in recombinant protein purification. Biomedical and pharmaceutical uses, where it is used for therapeutic protein production such as antithrombin 3. It is used in removal of contaminants such as unwanted proteins from biological fluids. It is also used in nucleic acid purifications like isolation of RNA, DNA, and other nucleic acids for genetics research. Specialized affinity chromatography, immunoaffinity chromatography, which involves purification of antibodies and antigens for immunological studies. Dye-ligand affinity, used to mimic biological substrates, purifying diverse proteins, enzymes, and pharmaceutical agents. It is also used in immobilized metal ion affinity chromatography, which purifies metal ions that contains histidine proteins. Thank you.